Good morning, everyone, and thank you for being here today. I'm excited to introduce our presentation on the role of fluid mechanics in pharmaceutical drug development. In this session, we'll explore how understanding flow dynamics can lead to more efficient drug design and manufacturing processes. I'll also share some insights and practical applications that can make a real difference in the pharmaceutical industry. Let's get started. Let's take a closer look at the overview of drug development and its economic prospects. The drug development process is quite complex and involves multiple stages. It starts with discovery, where potential drug candidates are identified. This is followed by preclinical testing, which involves laboratory and animal studies to evaluate safety and efficacy. Next comes clinical trials involving human subjects, which are typically divided into multiple phases to assess safety, dosage, and effectiveness. After successful clinical trials, the drug moves into manufacturing and finally reaches the market. This entire journey is quite lengthy, usually taking between 10 to 15 years to complete. And it's also highly expensive, often costing more than $2 billion to bring a single drug to market. Now, when we compare the economic aspects, we see some interesting improvements with newer methods. For example, the mixing time, which is how long it takes to mix ingredients, decreases from about 60 minutes without certain efficiencies to around 35 minutes with them. That's a significant saving in time. Similarly, yield efficiency improves from 78% to 92%, meaning we get more usable product from the same amount of input. Batch rejection rates also drop from 8% to just 2%, reducing waste and increasing overall productivity. And in terms of production costs, we see a reduction from 10 lakh rupees to 8 lakh rupees per batch. These improvements are crucial because they directly impact the overall cost, speed, and efficiency of drug manufacturing, which ties back to the broader theme of optimizing the drug development process and making medicines more accessible and affordable. Let's start with Reynolds number, which is a key concept in fluid dynamics. The formula is Rho equals rho times V times D, divided by mu. Here, rho is the fluid density, measured in kilograms per cubic meter, and it tells us how heavy the fluid is. The V stands for flow velocity in meters per second, essentially how fast the fluid is moving. D is the characteristic diameter, or the size of the pipe or channel, in meters. And mu is the dynamic viscosity, measured in Pascal seconds, which describes how sticky or resistant the fluid is to flow. Now, why is Reynolds number important? It's mainly used to determine whether the flow is laminar or turbulent. Laminar flow is smooth and orderly, while turbulent flow is chaotic and mixed. This distinction is critical because it affects mixing efficiency, reactor design, and the stability of suspensions. Understanding whether your flow is laminar or turbulent helps optimize processes and improve outcomes. Moving on to the continuity equation, which is simply A1V1 equals A2V2 here, is the cross-sectional area of the pipe or channel in square meters, and V is the flow velocity in meters per second. This equation essentially states that the mass of fluid flowing into a section must equal the mass flowing out, assuming there's no accumulation. Why does this matter? Because it ensures mass conservation throughout your system. Maintaining accurate flow rates is vital for dosing precision and for scaling up processes. If you change the pipe diameter, for example, the flow velocity must adjust accordingly to keep the flow consistent. So, both Reynolds number and the continuity equation serve as fundamental tools in understanding and controlling fluid flow in various engineering applications, helping us design better, more efficient processes. In this slide, I want to focus on how shear stress plays a crucial role in drug development processes. Shear stress, represented by the equation tau equals mu times do/di, helps us understand how fluid forces influence our formulations. Here, tau is the shear stress measured in pascals, mu is the dynamic viscosity of the fluid, and do/di is the velocity gradient, which tells us how quickly the fluid's velocity changes across a distance. Understanding shear stress is vital because it controls the mixing intensity during formulation. This ensures that our active pharmaceutical ingredients, especially sensitive APIs, are evenly dispersed without risking aggregation or denaturation. Essentially, by managing shear forces properly, we can maintain the stability and efficacy of our drugs. Now let's look at some key applications. First, in mixing and homogenization, shear stress helps us achieve a uniform dispersion of components, which is critical for consistent drug quality. Next, during crystallization, shear influences particle size and purity, both essential for ensuring the desired drug properties. Lastly, in coating and encapsulation processes, controlling shear affects droplet size and the formation of uniform films, which are crucial for controlled release and stability. So, in summary, understanding and controlling shear stress is fundamental across multiple stages of drug development, ensuring our formulations are effective, stable, and of high quality. Let's take a closer look at fluid mechanics in bioprocessing, starting with Bernoulli's equation. This equation is fundamental because it describes the conservation of energy in flowing fluids. It states that the sum of pressure energy, kinetic energy, and potential energy remains constant along a streamline. In the equation, P stands for pressure energy, measured in pascals. The term 1/2, ρ2 represents the kinetic energy per unit volume, where ρ is the fluid density and v is the velocity. The ρ term accounts for potential energy due to height or elevation, with g being gravity, which is approximately 9.81 meters per second squared. Understanding this equation is crucial because it helps us predict how fluids behave under different conditions, especially in bioprocessing environments. For example, maintaining the right pressure and flow rates is essential during sterile filling, pressure control, and filtration processes. These are key to ensuring product quality and safety. Now, moving to bioprocess applications, fluid mechanics plays a vital role across various stages. During fermentation, proper oxygen and nutrient distribution depend on controlling flow and shear forces to prevent damage to cells. Minimizing harmful shear is critical to maintain cell viability and productivity. In downstream processing, fluid mechanics helps us optimize filtration and centrifugation steps. Efficient flow control ensures we can separate target products from impurities effectively while preserving their integrity. Finally, in microfluidics, which is like lab-on-chip technology, precise fluid handling enables rapid screening and analysis. This technology allows us to work with very small volumes and achieve high-throughput testing, accelerating development and quality control. Overall, understanding fluid mechanics enables us to design better bioprocesses, improve efficiency, and ensure the safety and quality of biopharmaceutical products. So, to wrap things up, fluid mechanics plays a crucial role in the pharmaceutical industry, especially when it comes to mixing, transport, and formulation processes. Understanding these principles helps us achieve higher yields, reduce waste, and ultimately improve the quality of the products we develop. Looking ahead, there are some exciting directions we can take. For instance, using computational fluid dynamics, or CFD simulations, allows us to optimize design and scale up procedures more efficiently. This means we can predict how systems will behave before building them, saving time and resources. Additionally, integrating AI-driven flow control systems offers the potential for more precise and adaptable manufacturing processes. This can lead to even better consistency and efficiency. And finally, moving toward continuous manufacturing is a game changer. It offers a more cost-effective way to produce pharmaceuticals by allowing for seamless, ongoing production rather than batch processes. All these advancements tie back to our core goal: leveraging fluid mechanics to innovate and improve pharmaceutical manufacturing, making it more efficient, sustainable, and capable of meeting future demands.